Welcome everyone. With Swiss time precision, we get started at 5. So welcome to our webinar. My name is Marcus Mangold. I'm one of the co-founders of IR Sweep, and I'm the host of this webinar today. And it's my great pleasure to have Joachim Heberle as a speaker today, who will share his experiences on rapid reaction monitoring of proteins with dual-com spectroscopy. So let's get right into it. First, a brief outline. I will start in the first few minutes to give an overview over the benefits of dual-com spectroscopy and show what applications that dual-com spectroscopy can be used for. Then I will hand over the word to Joachim Heberle, who will give the actual body of the talk, where he talks about his experience so far with measuring with the Iris F1 dual-com spectrometer from IR Sweep rapid protein reactions. And then, very importantly, there's going to be a Q&A session at the end. So please, everyone, use the Q&A functionality of Zoom. There's a button at the bottom of your screen, and there you can type your questions, and we will make sure to answer them at the end of the webinar. Now, here you get to see a glimpse of the Iris F1, our dual-com spectrometer. And when we talk about our dual-com spectrometer, we mostly speak about 4 advantages that it offers, and those are high measurement speed, high spectral resolution, the high brightness of the source, and parallel multi-wavelengths acquisition. And I'd like to say a few words on those 4 advantages, why they are useful for measurements. Let's start with the high spectral resolution. Here you can see methane measurement, and the advantage that dual-com spectroscopy offers here is that you actually have a very very narrow emission lines of your lasers, and therefore you can measure with extremely high resolution your spectra. So the resolution that can be achieved is on the order of 10 to the -3 to 10 to the -4 wave numbers. This is mostly useful, of course, for gas-phase measurements, as for in the example here with methane. And if you are particularly interested in such applications, please look at our former webinars. 1 week ago, we had a webinar with Muriel Lepere from the University of Namur, where she's proven the high resolution of the spectrometer. And earlier, there was a webinar with Lukas Emnecker and Michele Cianella. The second point I'd like to mention is the high brightness of the source. And here on the center graph, you see what we mean when we say high brightness of quantum cascade laser sources. So on the y-axis of this graph, we have the optical emission power of the source as a per wavelength. And as you can see at the bottom, there's the global sources of the FTIRs, and then there are many orders of magnitude until you reach quantum cascade laser emissions. That is not only true for the quantum cascade laser frequency combs that we use in the Iris F1, but also for external cavity quantum cascade lasers. So that's a general advantage of using lasers. And why is this good? That is represented in the graph on the right. This helps you to work with very strongly absorbing samples. And this is now a point that is probably important for biospectroscopy, because you often work with samples which have very high absorption. For example, when you have a water background. So what is represented in that graph is how thick of a layer of some solvent you can transmit with the lasers that we use. And you see in most of the spectral ranges that is about 100 micrometer of water that can be transmitted. There is this little dip at around 1600 wave numbers, but even there, still about 20 micrometer of water can be transmitted, and that should be compared to what you can typically do with a global light source, where you're limited to something like 5 micrometer of water. And this is not only advantages to get stronger signals, but it also helps to for having easier sample preparation. Then the third point I mentioned is parallel multi-wavelength measurements, and I'd like to show you what we mean with parallel multi-wavelengths. So, as I said, we're using frequency comms, and frequency comms are not just normal single wavelength lasers, but it's lasers which emit many wavelengths at once. So a typical emission spectrum of such a laser is shown here in the center graph. You see it covers something like 60 to 100 wave numbers, and then it has several hundred emission lines, which are usually spaced with roughly 0.3 wave numbers. And all of those wavelengths are then measured in parallel. So that helps you if you want to measure multiple features at once. If you have complex samples, then you can get not only a single wavelength emission, but you can see a complete spectrum. And I think in the second part of the talk, we will see some example of how having a full spectrum helps to evaluate your data. Also, what is important to mention here is 60 to 100 wave numbers is, of course, not the full mid-IR spectrum. So the instrument is built in a way that you can easily exchange your laser sources. And that's what's shown in the picture on the right. Here you see the laser modules, which can be taken out, and the new laser module can be plugged such that you can access a different wavelength range. And talking about wavelength ranges, generally everything between 900 and 2200 wave numbers is accessible with QCL frequency comms. Some of the lasers we have directly on stock, and that's what is shown here. And very happily, I can announce that we have a new wavelength since basically this week available at 1740 wave numbers. I think also wavelengths which is useful for biospectroscopic applications. But please also don't hesitate to contact us if you need some other wavelengths in the range 900 to 2200 wave numbers. And now let me come to the fourth and probably the most important feature for the presentation of today, which is the high measurement speed. So when we speak about high measurement speed, I've shown you we have several hundred wavelengths at once, but how quickly can we see all of those wavelengths? And the answer is typically once per microsecond. So you continuously get every 1 microsecond spectrum, and that's why a typical measurement with the Iris F1 looks like the measurement that is shown here at the center. You have a coverage of above 60 wave numbers, and for this coverage, you get every 1 microsecond spectrum continuously for tens to up to hundreds of milliseconds. I can also say quite recently we have worked on pushing that time resolution a bit to the limit. That was in collaboration with the group of Markus Gerhardt at TU Kaiserslautern. And here you can see those measurements, which each of them represent a state of a sample that only lasted for 250 nanoseconds. So that is what is currently possible. The limit of the time resolution we see somewhere at around 100 nanoseconds of technology. So much about the overall features. Let me show you an overview of applications. You can see there's already a bunch of applications that have been shown with the Iris spectrometer. Starting at the bottom right, we have those gas analysis and high resolution spectroscopy things. Then there's also polymer chemistry, an application where people have shown curing of samples. VCD, vibrational circular dichroism, is something that was recently tapped into. And then maybe going through the top line, there's spectral electrochemistry, an application that has recently got quite some attention, where people follow the electrochemical reactions using the speed of the Iris F1. Then stopped flow is something where you're more interested into longer time ranges. So that starts more in the millisecond time range and looks at reactions that last for seconds or even minutes. Combustion diagnostic is a big topic, and of course, and that's why we're here today, protein dynamics. Now, if you're interested in learning more about some of the applications, we have a bunch of webinars. You see here, there have been people involved in those fields who reported their experiences on measuring with the Iris F1. And now, today, we get to have that talk of Joachim Heberle. And I'm very happy that he is willing to share his experience that he had. A few words about our speaker today. Joachim Heberle has been working in biophysics for most of his career. After a PhD in Berlin, he's been at the research center in Jülich, and then has been appointed full professor first in Bielefeld, and nowadays he's back in Berlin. And he spent most of his career not only developing but also applying time-resolved spectroscopy methods, and therefore I think he's perfectly well suited to talk about our spectrometer, and I'm very happy that he's willing to do that. And with this, I hand over the word to Joachim.
Thank you very much, Markus, for this very kind introduction. It's a pleasure for me to give this webinar here in front of a quite nice audience that I realized a few colleagues are there. Hi, John. Hi, Markus, and a few others are around there. So it's a pleasure to speak in front of you, although it's just here virtual. All right, let me just try to share my screen with you. Here. Okay. So here is the title of the presentation. I will talk about rapid reaction monitoring of proteins with the dual comb spectrometer that we purchased about, what was it, I think a year or a little bit more than a year from the company IR Sweep. It's called Iris F1, and you will see a photograph in a minute. Our interest in infrared spectroscopy comes from the fact that we want to answer the mechanistic questions of proteins, in particular membrane proteins. And if you know that about 80% of all drugs on the market actually act on membrane proteins, you see the biomedical relevance of these studies. There is other advantage, which is as compared to, say, fluorescence spectroscopy, that we can work label-free because we use the native vibrations of 2 or more atoms, molecular vibrations of molecules, and measure the absorption that is associated with these oscillations. The time resolution is basically unlimited up to the natural limit, which is the oscillation of 2 atoms versus each other of a chemical bond, and this is typically in the femtosecond time range. And a femtosecond is in the order of 10 to the minus 15 seconds. And time-resolved infrared spectroscopy has been applied in a great diversity of systems. And what is typically used in time-resolved infrared spectroscopy is pump probe spectroscopy, if you're interested in ultra-fast reactions, starting, as I just said a minute ago, in the femtosecond time range. And then you can go down over the picosecond and nanosecond. Some people achieve these days even microseconds by electronically linking 2 pump sources, 2 laser sources to each other. But this is a very difficult technique, still a difficult technique, I would say visible pump and infrared probe. Certainly it is more pricey than normal spectrometers that you can buy from a vendor. And the most prominent ones are certainly, and most experienced ones are FTIR spectrometers, where you can apply rapid scan spectroscopy, which has a time resolution in milliseconds. If you want to go higher, you need to apply step scan technique that brings you routinely to the microsecond range, and but also to the nanosecond range. That's possible. But these techniques, in particular step scan spectroscopy, require strictly a repetitive process. because you need to scan through the interferometer space and repeat the reaction that you're interested in always fully repetitively. Now, there are two new devices on the market that largely rely on the QCL, the quantum cascade laser, as the source of infrared radiation. And Markus already introduced the virtues of that. And this basically the major important aspect here is the high power of the QCLs. And either you put the QCL emission, which is natively a broadband emission, you put them into via an external cavity, the whole emission into one line, then you have a very intense emission, monochromatic emission, or you use the full width of the QCL emission and modulate then this with the help of this dual comb technique that is implemented in the IRIS F1 spectrometer. The IRIS F1 spectrometer has a time resolution routinely in the microsecond time range, but you can achieve, as Markus just alluded to, the nanosecond time range by new developments that they did. We have set up a external cavity QCL scanning type spectrometer, which is a home-built one, where we have an inherent time resolution that is limited by the detector that we have. So it's in the, say, 10 nanosecond time range. And depending on the memory of your oscilloscope, you can go down to the second range, and I will show you data. So this is about the technology. The system that we are interested in are proteins, as I said, and if you have a photosynthetic protein like those involved in photosynthesis or in vision, they come with a cofactor like the retinal that we have in our eyes that allows us for vision, is the cofactor that absorbs the light when we see. And this reaction is ultra fast. It starts already in the femtosecond time range and then goes all the way down from femtoseconds over many decades of time, even down to the millisecond time range for the rhodopsin reaction. Now, these are ultra fast reactions. They can induce in some systems, like in photosystems, in plant photosystems, electron transfer reactions. So they start roughly in the picosecond time range and can extend also into the millisecond time range. My group is very much interested in proton transfer reactions. They can be coupled also to electron transfer reactions, but not necessarily. And because the mass of the proton is larger than the electron, they are inherently a little bit slower than the electron transfer reactions, but can also span a very broad time range. And associated with these charge transfer reactions are then, and induced by them, are or can be protein conformational changes. And since there the mass is even higher that you need to move, the fastest of those is typically in the nanosecond time range, but again, it depends on the number of atoms that are involved. And some of these take seconds, even minutes is possible in biology for protein conformational changes. All right, now the spectrometer that we are using is a IRIS F1 spectrometer, and it comes with different modules. The first one that was on the market was this module 1, which has a center frequency of about 1200 wave numbers, and you have a tunability of plus minus 30 to 40 wave numbers, left and right. And here, the first application was in bacteriorhodopsin to look for the isomerization reaction. We have another module that allows us to go to a little bit higher frequency range, which is centered at around 1300 wave numbers. And there our project is on fluorinated compounds that you can find in Teflon pans if you want, right? But we are interested in the interaction of water molecules with these extremely hydrophobic surfaces. And module 3 is the emission of the module 3 is at around 1650 wave numbers, so ideally suited to probe the amide 1 mode of the protein backbone, so the carbonyl stretching vibration of the protein backbone. And this is indicative for structural changes in in the whole of the protein. And then finally is the one that we just purchased not so long ago. This is the module 4 with a center wavelength at around 1760 wave numbers. And there we can probe protonation dynamics, because as you will see in a minute, we can make use of the carbonyl stretching vibration of aspartic acid or glutamic acids and look at their protonation or deprotonation reaction. So these 4 modules can be all implemented in the IRIS F1, and this is what we have available in my lab. Now here is the model system that we are using for our initial studies. This is bacteriorhodopsin, which you see is a membrane protein on the left side here in purple, and this pumps protons across the membrane from the inside to the outside in this cartoon, which establishes a proton gradient across the membrane. And this eventually drives ATP synthesis through this ATP synthase here. And ATP is adenosine triphosphate, the fuel of all, basically all cells, not only in our body, but even in plants and in bacteria. Okay, so this bacteriorhodopsin is a photosynthetic system. It has a ground state absorption at 570 nanometers, which gives rise to a purple color, which you see here in the background. So this bacteriorhodopsin ground state is purple if you have it in solution. And if you excite this by a short laser pulse, then bacteriorhodopsin undergoes a cyclic photoreaction. And it passes various intermediate states named J, K, L, M, N, O, until it eventually returns back to the original state. So it's very convenient because you can average, of course, many reactions, because bacteriorhodopsin, once it has been excited, it returns to the original state back, and you can re-excite the very same molecule without moving or whatever. What is also depicted in this graph is the time range that this photocycle covers. It starts out here in the femtosecond time range, picosecond, microsecond, millisecond, until it comes back. So you see there's a huge time range that is covered, and with the IRIS F1, we can start investigating the photocycle, let's say from the K to L transition. So when the K decays into the L, this is where we start our study, and this then, the rest of the reaction can be traced with this. There are reference spectra already available, and you see here, these are data from that many people have already recorded. I just show the data from my lab here, which are the different spectra of the K minus BR, different spectrum of the K intermediate, the L intermediate, M, N, O, and so forth. And you see also the frequency ranges. Here, the CC single bond stretches the fingerprint region that is indicative for the retinal isomerization. The CC double bond stretching vibration of the retinal chromophore is here in this frequency range. Then the amide1 is here in this frequency range at around 1650 wave numbers. And then above 1700 wave numbers, the carbonyl stretching vibration of aspartic acids and glutamic acids. And these are basically the 4, well, actually 3 of those are the 3 frequency ranges that are displayed here come with laser modules that we purchased. Except for this one, we don't have that, but there is our module number 2 that covers this range here, which is here for bacteriorhodopsin, not so interesting. Okay, now the pioneering study was published here from the lab of Tilman Köttke from Bielefeld University in collaboration with the people from IR Sweep. So you see Marco's name here. And they collaborated and measured bacteriorhodopsin in this frequency range between 1240 and 1180 wave numbers. And we see here these fingerprint modes that appear in this difference experiment. And those are indicative of the fact that the cofactor is isomerizing. And you see here a comparison between the step scan data on the right and the IR Sweep data, or the IRIS data. And they are in excellent agreement. It depends, of course, on the number of averages that you do to achieve the best signal to noise. I don't want to discuss this any further because this has all been published already 2 years ago. Now this was in this frequency range because at that time there was no other laser module available. Now we purchased then a laser module for the amide 1 region between 1680 and 1620, and you see a time-resolved experiment that is shown here in this 3D plot. The time is now shown now on a logarithmic time scale, starting here from 1 microsecond all the way down to 0.1 seconds. And this is the frequency range, and this is the absorption change. The global fitting, because this is a kinetic data, identifies 3 intermediate states that are related to the photoreaction of bacteriorhodopsin. And those spectra are in excellent agreement with published FTIR data, StepScan FTIR data, of course. These were, I guess, 3,000 repetitions. In an FTIR experiment, you would need certainly something like 100,000 repetitions to achieve this signal to noise here. Here is a comparison now towards single shot experiments. It's not a comparison, but just to show you these experiments. This is 3,000 shots measured with the iris f1 in the amide 1 range. This is in the time range of the M intermediate. This is about 200 microseconds after the initiation of the photoreaction. And you see these different spans here very clearly here at 1670, 1660, and around 1635 wave numbers here. And at a later stage, at around 2 to 3 milliseconds, the NO intermediate appears. You see how these bands start to decay. And we can follow the kinetics of the band that is shown here at 1670 wave numbers. So this is this band here, plotted over the logarithm of time. And you clearly see with the time resolution here of 5 microseconds that it decays here until it has its minimum reached here. This is the point of the largest conformational change of bacteriorhodopsin until this recovers back to the original state. And we measured here until 10 milliseconds. Of course, the photocycle goes a little bit longer, but not too much longer. Now, these data were recorded with 3,000 shots. And now let's compare this to a single shot experiment, which is possible with this iris f1 device. And these are the raw data. There was no data treatment. Well, no, there was some data treatment, but this is what we call a raw spectrum. And this is done at the same time after the laser flash like the one on top. So you can directly compare these two, and you see, of course, there's more noise here in these spectra, but overall spectra look similar. And here in the later stage where the logarithmic averaging is stronger, you can see very clearly that these are comparable. The difference is simply these are 3,000 shots, and this is a single shot experiment. And if you look at the kinetics at 1670 wave number, so again, let's say this band here, and we follow the kinetics here, then this looks a bit noisy, but I will show you later that data treatment and data analysis shows that this is very similar to these kinetics here. Okay, so single shot experiments are possible in this frequency range with iris f1 spectrometer. Now, we will go now to a different frequency range because, as I said in my introduction, we are also interested in proton transfer reactions. And for that, we make use of the carbonyl stretching vibration of the amino acids aspartic acid or glutamic acid when they are protonated. So only when they are protonated, when there is a proton, the CO double bond emerges, which gives rise to a infrared absorption band in the range above 1700 wave numbers, so in this frequency range. When this loses a proton, then it loses also the double bond character here, and the band is gone, and this gives rise in a difference experiment to a negative absorption, whereas positive absorption corresponds to the protonation of such an amino acid. And of course, such a 3D plot can be also represented in the form of a heat map. So here's the frequency now. It's the same frequency axis than on the left. On the x-axis here is the logarithmic time axis from 5 microseconds to 20 milliseconds. And you see in this heat map the positive band, which is this positive band here. So this has a peak maximum at 1762 wave numbers, which is maximum at around 200 microseconds or 400 microseconds after the laser flash. And there is here a deprotonation, so a negative feature, which is this one. All right, so this is for the presentation, but as a spectroscopist, you want to see spectra, and this is shown here. These are different spectra after certain times, 10 microseconds, 200 microseconds, 3 milliseconds, 8 milliseconds. These are step scan data, just for reference. It's not iris data. Just to show you the fidelity of the data, but comes from step scan data. And the assignment has already been done. This positive band is from a residue with the number D85. This band is due to D96, D115, D212. This is just for the bacteriodopsin experts. It only makes sense when you put this information into a structural context. And here is the X-ray structure. Bacteriodopsin pumps protons from the cell interior, which is here, to the outside, which is here. This is the extracellular surface. And now we can make use of this information from infrared spectroscopy and put arrows here in that delineate the proton transfer pathway in bacteriodopsin. So one is, for instance, the protonation of the D85 here, and this gives rise to this signal. And this appears with a time constant of 50 microseconds. So this is all known, and many labs in the world contributed to this very detailed knowledge. The image that is shown here is from a publication that, if you want to look it up, it's more than 10 years ago already. Okay, but now let's come to our iris spectrometer. And what you see here is the data between 1780 and 1745 wave numbers after 3,000 shots. So you see clearly a positive band here after 200 microseconds. This band also shifts, like what we would expect also from the step scan data. And we see also the kinetics here, 3,000 shots. Kinetics are very flat here, but just to remind you, what is the scale here of the absorption change, right? So this is less than 1 milliod, so to say. So the noise is in the order of 10 to the minus 5, or say 5 times 10 to the minus 5, with 3,000 shots. But now you're interested maybe in what happens with less shots. And here is now 10 shots. And even with 10 shots, you can see this band, and you see it even shifting. And no wonder the kinetics are very noisy here. But with this LDA analysis, which is a lifetime distribution analysis, you can clearly say that there is some kinetics in there that resemble the highly average data. Now, let me compare now, or make a critical comparison of the dual comb technique with the EC, with the external cavity QCL technique that we have set up. And this is published in the publication, or the reference is shown here at the bottom. And I think Bernd Schulz is also in the audience here. Here on the left side is the EC-QCL. It's a scanning type because we have a monochromatic light source. And for each repetition, we have to scan it to a different frequency output with the help of the external cavity. Spectral coverage is about 100 wave numbers and for one laser, and you need more lasers to cover a broader spectral range. The time resolution is nanoseconds, which is, as I said before already, higher than the microseconds from the dual comb, but as Marcus already said, this can be improved. Here in the dual comb spectrometer, spectral coverage is in our hands 60 wave numbers, so it's a little bit narrower than that with the EC-QCL. About the time range already discussed, you will see that for the EC-QCL, we need a repetitive process still. But for dual comb, we can do this, we can apply the technology on non-repetitive reactions. Now here is the data comparison of the home-built EC-QCL, that's this one here, as compared to the IR sweep Iris F1 device that is shown here. And here are the data in direct comparison after 3,000 repetitions. No wonder data are identical, more or less, based on this comparison. You also see under more or less comparable conditions that the noise in the iris data is a little bit higher than in the ECQCL, which is something we can discuss. Kinetics is clear here on the right side. Is the time resolution with the ECQCL is nanoseconds. That's why it starts here. The dual comb is in microseconds. That's why it starts here. But it's the same time axis. I plotted this on purpose on the same temporal axis just to show here we have a higher time resolution. We can extend this also to slower times. This is now also improved with a dual comb spectrometer, as Marcus already alluded to. Now, the most important part is now what happens if we go to single shot experiments. This, of course, is with ECQCL not possible because you have to scan through the whole spectral range with the help of the external cavity. It is possible with the dual comb spectrometer, and you see it here. In green are the raw data. This is the after about a millisecond recorded. And if you do the lifetime distribution analysis, then you can even improve here the spectra or the spectral response here. So this really is I would say remarkable for a single shot experiment achieved with this device. Kinetics are shown here on the right side. Of course, you can record kinetics. This is a single shot kinetic of the ECQCL, but on 2 different wave numbers. So these are 2 different experiments. And here the LDA analysis, which is plotted in black here, shows also that the kinetics recorded by a single shot experiment with the iris F1 resemble the green one here, right? Of course, the noise is higher because we have per frequency much less photons than in the ECQCL for sure. Okay, this is what I wanted to share with you. In the end, let me just acknowledge my group, and this is a very nice representation that was painted by some artist member of my group. The persons mostly involved in this is Louis Schubert here. He's also in the audience, and you see him with his baby here. He calls it not Iris, but he calls it Irene. That's his girlfriend, so to say, when he's in the lab. David contributed with the LDA analysis, which turned out to be extremely useful for single shot experiments. And Pitt contributed with ECQCL data that were taken on the very same sample under more or less identical conditions. In the end, I would also like to acknowledge the financial support by the German Research Foundation, Deutsche Forschungsgemeinschaft, within the SFB 1078, because this provided the money to purchase the first spectrometer from IR Suite, because obviously we were the first customers to have this very nice spectrometer. So with this, I can conclude, and thank you for your attention. I'm looking forward to your questions.
Thank you very much, Joachim, for that nice presentation. Let me share my screen again. So it's time for questions and answers. We have some questions already here. One that came in is, how fast can you measure with dual comb spectroscopy, nanosecond time resolution, and what is the spectral resolution then for 816 wave numbers? So the speed, I think we've seen that somewhat in the graphs. Maybe you, Joachim, can comment on that. Right.
No, I think this is exactly for you, because it's we had just a 1 microsecond experiment, right? And you have improved the time resolution in your experiments with the Gerhards group.
Yeah, so you can go down to faster time resolution. So standard is 1 microsecond that works off the box with some tuning. You can get down to 250 nanoseconds is what we have shown. Maybe for the experts in the audience, what limits your time resolution is kind of how many of your optical lines you want to measure in the radio frequency bandwidth that you have accessible. And therefore, one way to even further improve this time resolution is if you take shorter lasers, then you have less emission lines per wavelength, and therefore you have to measure less wavelengths to cover the same, say, 60 wave numbers, and like that you could get down to even faster time resolutions. What we see as a reasonable limit for the technology is around 100 nanoseconds. Maybe also talking about the other end of the time axis. Now the spectra that you've shown, Joachim, all ended around 16 milliseconds. Actually, with the highest time resolution, you can go up to something like 250 milliseconds at maximum. So we already get close to the range of 1 second. If you care for even longer processes, then you can actually do a different measurement mode where you have less time resolution, roughly 1 millisecond time resolution, but then you can actually measure for as long as you want. Another question came in. Can you apply dual-com spectroscopy also for non-repeatable processes, which you haven't shown yet? Because bacterial rhodopsin is obviously repeatable, and StepScan and external cavity QCL can be applied as well. So that is probably a question for you, Joachim.
Yeah. So that's why maybe I go back to my PowerPoint presentation and just show you a backup slide. So here is, for instance, it's not a fully irreversible reaction, but it's a reaction that takes place, takes very long to recover. And this is also retinol protein, so it's also light driven. It's channel rhodopsin. So this channel rhodopsin is a protein which is not a proton pump, but it opens an ion channel, and ion channel opening can lead to nerve activation in optogenetic experiments. That's why there's some relevance of this work for neurobiology to understand functional mechanism of channel rhodopsin. And what you see here, these are time resolved data recorded on channel rhodopsin 1. And you see here P1, this is the first intermediate of the photocycle of channel rhodopsin 1 after 75 microseconds, and the black one is after 1 millisecond of the P2 intermediate, which is the open state of this ion channel. And the cyclic reaction of this protein is, what is it, around 10 seconds is the time constant for the last process. So here you can repeat the experiment only, let's say, every 20 or 30 seconds. And so averaging is very hard, but I would like to go back to the experiment that I've shown you before, which is the single shot experiment. And the single shot experiment on here. Oops. So here is the single shot experiment. So this is bacteriorhodopsin, but we did not, we excited it only once. So it's truly a single shot experiment, and you can see this works. Even at high time resolution in the microsecond time domain, and this is here in the millisecond time domain. So you can do these experiments, and we are about to apply this to receptor proteins, for instance, yeah, where you have like a GPCR, a G protein coupled receptor that binds some agonist or antagonistic molecule that you release by a short laser flash via a caged substrate, and then this binds to this receptor, and then you might be able to trace this reaction. I hope that answers your question sufficiently.
Okay, thanks for the answer. There is probably very much related question from Jakob Hayden. He writes, question to Professor Heberle: can you comment on the significance of single shot measurements for your research?
Yeah, right. So this is connected to, I just can continue to what I just said before. I think this would be really great for time-resolved infrared spectroscopy who are interested in say, mechanistic studies on completely different proteins that are of extreme relevance, like the GPCRs, the G protein coupled receptors. If you can do time-resolved experiments like much in the same way as we do with inherently light-sensitive proteins, and can apply the very same technology like time-resolved infrared spectroscopy on systems that just make one catalytic turnover, right? Like an enzyme that gets an educt and then makes one chemical reaction, and then there is the product. So you cannot repeat this reaction, or not very often. You can do a stop flow with limited time resolution. But here, this offers truly the possibility of working on proteins whose reaction is irreversible. And our interest is our membrane proteins. And these are very difficult to handle also. But this is the challenge that we are facing, but we are happy about challenges.
Great. John Kemmins also has a question. He writes, thanks for the illuminating seminar, Joachim. Did you see any effects of the rather high infrared intensity on the sample?
Yeah, very good point. Thank you very much, John. These are, the good part about this is infrared spectroscopy as compared to visible spectroscopy. Of course, the photons are of much less energy that we are using for measuring. Still, the sample is a stationary sample. So we leave the sample more or less in the measurement beam, but we tested this, and that there is a slight heating with the ECQCL. So we did it first with the ECQCL, with the extra cavity, which has a higher power, but squeezed into a very narrow band, and compared it also to the more broader emission of the dual comb spectrometer. And if there is at all a temperature increase, it is less than 1 degree Celsius in our hands.
Okay, good. So they don't burn away. The next question is, can you also determine secondary structure in proteins like that and do, for example, stress tests with temperature increase?
Right, yes. That's also a very interesting point, and it relates also to our science, but we don't do time-resolved, say, unfolding or folding experiments by temperature jumps. This is something that, for instance, my colleague Karin Hauser in Konstanz is doing. So they use a laser that makes a temperature jump and then follow, say, protein structural responses to that. And this is extremely interesting, and sometimes we touch this area because you can imagine if you use visible light here for a retinol protein, not all of the energy that you dump in the protein is used for the chemical change in the protein, but some, and actually quite a lot of the energy is just goes to the solvent environment, so to say, as heat. So we have to deal also with heat, maybe artifacts or whatever. It's something that we always need to consider in the interpretation of our data.
Okay, thanks. Then another question here is, in FTIR experiments with the glow bar source, one is typically limited to 10 microns or less in H2O and 50 microns in D2O. Is it possible to use much larger path lengths in the Iris F1? So maybe I can start answering this question. Yes, it is, because we have a lot more power. At the beginning of the talk, we've shown a plot about it, and it's on the order of 4 times larger. But maybe now let's anyway forward the question to you, Joachim. Did you actually, are you using longer path lengths in your lab because you have this more power?
Yes, yes. So in particular, when it comes to the carboxylic range above 1700 wave numbers, so here we can use, say, optical thicknesses of, say, 50 micrometer, because the background absorption is not so strong from the water. But also for the amide, here we use typically 10 microns because, and this is very important for proteins, because the amide 1 mode of proteins also absorbs at 1650 wave numbers. So we have the absorption of the amide 1 plus the absorption of the water that we have. So the optical density of the sample is very high, and therefore it's extremely useful, I would say. When it comes to proteins, there's an additional positive effect when you use such a spectrometer like the dual comb spectrometer that is able to penetrate highly absorptive samples like a protein sample in water. Yeah.
Okay, so you're actually using that power. John Kenneth has another question. He asks if you tried to use the spectrometer in ATR mode. Did you ever do that?
We are currently setting this up.
Okay, great.
My co-worker Pitt Langner, who is I think also in the audience, that's one of his projects.
Okay, good. And maybe I can also say something about that. We did a bunch of measurements in ATR mode. There's also some application notes on our website, and for example, for curing measurements, but also using those miniature ATR crystals from Irubis, where basically only small grooves are used or are formed on the chip, and then those are used as ATR crystals. And in general, it always worked fine also with ATR crystals.
Mm-hmm. We also use those.
Sorry. Then last question from Katie. Very good presentation. Thank you very much. May we ask, how strong is the power on the sample? Does it induce heating? How big is the IR spot on the sample? So we've had part of the answer, but how strong is power and spot size? Do you know those details?
Oh no. For this, I either have to refer to you or to my co-worker, Louis Schubert.
Okay, so I can, of course, not speak about what it looks like in your lab, but the standard spot size or the beam diameter in the sample chamber is around 3 millimeter. So if that is not modified, then that is typical. And the power, that depends on how strongly your sample absorbs. At the end on the detector, you want to have roughly 1 milliwatt. So if you have hardly any absorption in your sample, then 1 milliwatt of power incident is enough. If, of course, your sample absorbs 99% of the light, then you would turn that up to 100 milliwatt. Actually, the upper limit is typically 100 milliwatt, depends on the laser that you have, but up to 100 milliwatt is usually possible. Okay, and I think we've gone through all the questions that are currently here. Thank you very much to the audience for the active participation, and thank you very much to Joachim for the great presentation.
Thank you.
And hear you again soon.
Mm-hmm. Okay, thank you. See you. Bye.
Goodbye.